Good day! This time, we will be tackling about the molecular genetics of bacterial plant diseases.
That's right. Now let's know that bacteria enter the intercellular spaces of plants through wounds or natural openings such as stomata.
Therefore, bacteria do not need to penetrate the plant surface but they must have ways to adhere.
The attachment requires three components: a glucan molecule, the synthesis and export of which requires three genes for the synthesis of cellulose,
and the ATT region of the bacterial genome that contains several genes for attachment.
Moving forward, secretion systems are essential pathogenicity tools for bacteria because they make possible the translocation of bacterial proteins and other molecules into host plant cells.
And there are 5 forms of secretion systems that are recognized on the basis of the proteins that form them.
Let's start from the type 1 secretion system, which is present in almost all plant pathogenic bacteria and carries out the secretion of toxins such as hemolysins, cycholysins, and rhizobiosine.
These toxins consist of ATP-binding cassette proteins and are involved in the export and import of a variety of compounds through energy provided by the hydrolysis of ATP.
Moving on to the type 2 secretion system, which is common in gram-negative bacteria and is involved in the export of various proteins, enzymes, toxins, and virulence factors.
Next is the type 3 secretion system, which is the most important in terms of pathogenicity of the bacteria in the genera of Pseudomonas, Xanthomonas, and Ralstonia.
The primary function of type 3 secretion system is the transport of effector proteins across the bacterial membrane and into the plant cell.
Yes, genes that encode protein components of the type 3 secretion system apparatus have a 2/3 similarity at the amino acid level, and such genes are called hypersensitive response conserved genes.
Now, for the type 4 secretion system, transport of macromolecules from the bacterium to the host cell occurred here.
The proteins transferred are very similar to those responsible for the mobilization of plasmids among bacteria.
And lastly, the type 5 secretion system autotransporter is found in Xylella and Xanthomonas and contains genes that encode surface-associated adhesins.
Moving forward to pathogenicity of bacterial enzymes that degrade cell walls.
Plant cell walls are composed of 3 major polysaccharides: the cellulose, hemicellulose, and pectins, and in woody and some other plants there is linin.
The number of genes encoding cell wall degrading enzymes varies greatly in the different plant pathogenic bacteria.
Pectinases, for example, are believed to be the most important in pathogenesis as they are responsible for tissue maceration by degrading the pectic substances in the middle lamella and indirectly for cell death.
In addition, there are global regulatory systems like the quorum sensing system, so as to maximize the activity of the main enzymes.
And because of the large number of pectinases involved, disruption of the gene encoding any one of the enzymes is not sufficient to stop cell maceration.
Now, let's talk about the bacterial toxins as pathogenicity factors.
Yes, toxins have been known for a long time to play a central role in parasitism and pathogenesis of plants by several plant pathogenic bacteria.
Pseudomonas syringae, for example, are primarily associated with production of the phytotoxin coronatine.
The bacterium, along with its pathobars, produces several phytotoxins, including syringomycin.
Next is the extracellular polysaccharides as pathogenicity factors.
That's right! Extracellular polysaccharides play an important role in pathogenesis of many bacteria by both direct interventions with host cells and by providing resistance to oxidative stress.
In the bacterial wilt of solanaceous crops caused by Ralstonia solanacearum, extracellular polysaccharides 1 is the main virulence factor of the disease.
EPS likely causes wilt by occluding the xylem vessels and by causing them to rupture from the high osmotic pressure.
It's true, but now let's talk about the mechanisms of bacteria sensing plant signaling components.
Agrobacterium tumefaciens, for example, has a two-component regulatory system that senses and reacts to the presence of susceptible cells.
Moreover, lipopolysaccharide components of the outer cell wall of gram-negative bacteria also play a role in the pathogenicity of Erwinias.
Proof of this is provided by the activation of pathogenesis-related proteins such as glucanases in the infected plants.
And the fact that disruption of the lipopolysaccharide gene in the bacteria reduces their virulence and that protein lipopolysaccharide complexes from bacteria inhibit the hypersensitive response.
The peptide methionine sulfoxide reductase also, which protects and repairs bacterial proteins against active oxygen damage, is essential for the expression of full virulence of the bacteria.
Another is the HRP genes that encode proteins called harpins or pilins are used to make a type 3 protein secretion system that is used to deliver AVR proteins across the walls and plasma membrane of living plant cells.
Wow! Plant pathogenic bacteria have a variety of strategies to infect and invade in the environment as well as in their host plant.
That's right! Thus, the development and employment of management approaches to overcome and suppress phytopathogenic bacteria, which includes mitigating their infection strategies, is imperative to global food security.
This is Gidel M.V.U. Vicente.
And this is Julina Inardicido, your host for today's Microworld podcast.
That's all for today, listeners.
Have a good day!